Please come in, take your seats. We are about to begin our conference. Thank you for coming to Gliwice, especially at this dark season of the year. But I am sure that those 2 days will be bright and warm, enlightened by the light of science and warmed by the joy of our meeting. So, tak, ale my poprosimy.
I would like just to make tradition to start our conference, because I was opening it for 27 years. That's the second time. And I would like just to remind that this is conference 27th, yes, 28, 28 conference, which is annual, and that we try to make it every time interesting. And it is organized from the very beginning by the Association for the Support of Cancer Research. This is the non-governmental organization, and Institute of Oncology and Technical University, Silesian Technical University. From the very beginning, these 3 units were organizing this in different conditions. Last years we have this nice book. So this is from my side just to remind. And I think that I will give the now the president of the association and others for just continuing opening.
We would like to, at the beginning, invite to the front our very important person, our host, our generous supporter. The host and supporter of the conference is Silesian University of Technology. And I would like to invite Professor Bożena Skołd, who is Vice Rector of the university.
I would like to invite Professor Sławomir Blamek, the director of Gliwice branch of the Institute of Oncology. As you have heard, the Institute of Oncology is one of the main organizers. Welcome.
We would like to invite Professor Michał Mikula, who is Deputy Director for Science of the National Research Institute of Oncology in Gliwice. Our, I would say, mother institution.
Yes, and we invite Mrs. Katarzyna Kuczyńska-Budka, the mayor of Gliwice City, who is also our supporter and honorary patron. Welcome. Yes. And last but not least, we ask here the family of Professor Mieczysław Chorąży, yes, Mr. Tomasz Chorąży and Mrs. Iwona Chorąży-Kapitońska. Welcome. We have many events devoted to commemorate Professor Mieczysław Chorąży, and we always welcome the family of our mentor.
Okay, so I would like to ask our host, Professor Skołd, just for few words to participants.
Thank you very much. Ladies and gentlemen, guests, I would like to welcome you warmly at Silesian University of Technology. Unfortunately, our rector can't be here at this moment because he opening another conference here, and just over this wall. For us, it's very important to be here because we are beneficial of your results. What we are doing is important not only for science, but especially for us, for people. Thank you very much for being here. I wish you a nice day and very good result. And thank you for inviting me here. Thank you.
Thank you.
Thank you.
And we ask Professor Sławomir Blamek, our director.
Good morning, everyone. It's a great honor and a privilege to welcome you here to the 28th already Gliwice Scientific Meeting organized by National Research Institute of Oncology in Gliwice, Salesian University of Technology, under the auspices of Association for the Support of Cancer Research. I'm very proud seeing you here and to our international guests. It's a great privilege to host you here. Well, the fact that you are here, you are sharing your knowledge, your achievements, shows that the science truly has no borders, and together with fruitful cooperation which lasts for so many years, we can push the borders with the boundaries of science and learn new things and achieve new goals that we have in front of us. So I don't want to make this talk too long. I'm very happy also, I'm now referring to all participants, that you are so numerous, you are here, and I encourage you to actively participate in discussions and to challenge existing paradigms, to ask our distinguished guests questions, to seek for cooperation, because mutual effort can result with great achievements. I wish you a very fruitful conference, and again, thank you for being here.
Thank you.
Good morning. Hello, everyone. So this is my third conference here in Gliwice as the Deputy Director for Science, and when I always come here, it's a great pleasure to meet all of you, all the faculties and students. Although my previous presence here was for 1 day only, this time I'm staying for 2 days because this conference is important for the Institute. And this specific one is created under the umbrella of the omic knowledge. I am old enough to say that the omic has been born about 2 decades ago. And I was PhD student actually here visiting Gliwice because the government at that time decided to spend some funds to the new technologies, which at that time was the, I would say, proteomics at its infancy and expression arrays. So we got a huge grant to study colorectal cancer, and we got together here in Gliwice to decide which way to go. And it was striking that we, the biologists, physicians, mathematicians, biostatisticians, got together to embark on this journey, because we are creating the data. The data was uninterpretable at that time, and all those people from the University of Warsaw, from this University of Technology, were starting to, beginning to learn how to approach these large data sets. So this was the time when the omics started, and today's conference is about the achievements of the technologies and knowledge. Today we try to combine all those technologies together, but it won't be possible without the cooperation of different disciplines. So please appreciate that we are taking the knowledge of the previous generations of scientists that built the knowledge, that built the technologies, and today we can enjoy doing fun science. So have a great time in Gliwice. Don't be shy. This is to the students. Come over, let's talk, because this is about you. You are the future of science and medicine. A great day at this conference. Thank you very much.
Yes. And now we ask the mayor of our city, Professor Mrs. Katarzyna Kuczyńska-Budka, to welcome our participants.
Dzień dobry Państwu. Ja jako humanistka powiem po pierwsze, że jestem niezwykle dumna, że to miasto Gliwice jest miejscem, gdzie nauka ma należne miejsce i gdzie nauka jest niezwykle ceniona. Sam fakt, że Narodowy Instytut Onkologii jest umiejscowiony tu i że tutaj dzieją się wielkie rzeczy, jeśli chodzi o naukę, a tym większe, że to chodzi o to, że to nauka jest w służbie pacjentom, to miastu nie wypada nic innego jak wspierać, pomagać i mówić wokół właśnie o tym. Raz jeszcze dziękuję organizatorom, że przed wielu laty rozpoczęty cykl konferencji naukowych dotyczących zawiłych kwestii onkologii, ale tak ważnych dla tych wielu pacjentów, którzy przyjeżdżają tutaj w Gliwicach i że te dyskusje toczą się właśnie tutaj. Bardzo Państwu dziękuję, kibicuję, cieszę się, że na sali jest wielu studentów, bo to świadczy tylko o tym, że faktycznie w Gliwicach przyszłość to są ośrodki naukowe, to jest to, o czym Państwo debatują i co za chwilę będzie miało wymierne przełożenie na to, w którą stronę pójdzie medycyna. Bardzo dziękuję i życzę owocnych obrad.
Thank you very much. And now it's time to announce the results of the competition for the fellowship named after Professor Mieczysław Chorąży. Professor Mieczysław Chorąży was an eminent scientist and a great man. He was our teacher and our friend. It is already 3 years since he has passed away, but we still miss him. So this was our idea, supported by the Association for the Support of Cancer Research, to organize this competition. Our aim is to distinguish young scientists who are as much devoted to science as Professor Mieczysław Chorąży was, and who seem to follow his paths, life paths, also in other aspects. So this year the competition was very strong. We had 22 candidates, and I would say that the top 10 were really outstanding. So our scientific committee had a hard work, but finally they managed to find the best person, yes, because the winner is only one. So this year, this scholarship goes to a person who is working here in the Silesian University of Technology. This is Dr. Engineer Marcelina Jureczko. Congratulations. Can we ask? Yes, Marcelina. So you will get the diploma from the family members of Professor Chorąży. So congratulations. A photo, please. Come here. Okay. Thank you. And don't forget that your duty is to present your work shortly tomorrow in the afternoon session, and we welcome everybody to listen to our winner. Congratulations.
maybe in short, I've got the privilege to meet Professor Chorąży in person, and it adds the personal touch to this awardee. And I hope, and I just feel that I just gained the new energy to conduct further research. Thank you very much.
That's right.
Yeah.
Thank you. So we should assume that formally our this year conference is open. We can go to scientific content, and I thank you for our VIPs. I think that this scientific contents would be easier to digest, not from this from the front of the saal, but from the rest of the room. Okay, so Professor Chorąży passed 3 years ago. And to memorize him as a great researcher, we decided to initiate 2 activities. First, this is scholarship for young researcher. We just approach laureate of this year event. And second event is the lecture of in memory of Professor Mieczysław Chorąży, who is delivered just as a first event during Gliwice Scientific Meetings. Well, we try to invite as our lecturers excellent scientists, not to mention a person who provided this lecture last year, Dr. Victor Ambros, this year became a Nobel Prize laureate in the physiology and medicine. And this year we decided to invite to provide this lecture Professor Theresy Whiteside. Just a few words about Theresy. Professor Whiteside graduated from Columbia University in New York in 1967. He's associated with University of Pittsburgh from 1973. Since that time, she became professor of pathology, professor of otolaryngology, and professor of immunology. He's recognized expert in the field of molecular and experimental immunology. I said recognized. 60,000 quotation of her study just inform you what means highly recognized researcher. I would also like to mention that Theresy has Polish roots. She was born some time ago in Katowice. Okay, so Theresy, Professor Whiteside, I say Theresy because Professor Whiteside is our friend, so I feel that I can use your name. Professor Whiteside, please, floor is yours.
Well, I speak very loud, so I hope I don't have to hold the microphone. But can we use this one? Okay. All right. So. Nope. Doesn't work. Okay. First of all, I want to thank the organizer for inviting me to talk to you about my favorite topic, tumor-derived exosomes. But I also want to let you know that I feel very honored to give this lecture in memory of Professor Mieczysław Chorąży. And again, thank you for allowing me to have this privilege. Perhaps you all ought to revise the title of my talk slightly in your, in whatever you've got, because I'm going to talk about exosomes and their role in human disease and health, and tell you that they really are ubiquitous intercellular communication system. So we'll begin by very brief introduction, what are exosomes? And they are extracellular vesicles. They are surrounded by a double lipid protein membrane. Their surface is decorated by a variety of molecules, which you all know. Some are immunosuppressive, some are stimulatory protein. In the lumen, these vesicles carry a variety of cargoes, including proteins, nucleic acids. The proteins are active, they are enzymes, they are cytokines. So very rich content, which to a large extent sort of duplicates the content of their mother cells, cells from which they originate. These are very small vesicles. You can't see them under ordinary microscope. You have to use electron microscope. In cryo-electron microscopy at the bottom of the slide there, you can see distinctly this double membrane that surrounds each vesicle and a little bit of corona of proteins that are surrounding each vesicles. Corona is usually composed of components of plasma or serum or fluid in which these vesicles are found. You can also appreciate that the variety in their sizes. Now, how do exosomes are formed? So biogenesis. It's very interesting. This is a cell which is decorated by some proteins there, and by virtue of endocytosis, these proteins end up in the endocytic compartment in early endosomes, and then in late exosomes, there is this funny invagination process going on where the proteins that were originally inside the early endosome become transposed, if you wish, by this intraluminal invagination, so that they are now on the surface of the little vesicles inside the late endosomes, which the late endosomes fuse together, become multivesicular body, and multivesicular body goes to the surface of the cell membrane, and exosomes are released into intercellular space. And these vesicles now look, as you see, a little bit, they look like the original mother cell on the surface, and they carry information disseminated through entire body. So this process of formation, vesicle biogenesis, has been studied extensively. It's a complex process, and just to show you that we can actually visualize the exosomes being formed in tumor cells in this case, and this is multispectral analysis in tissues of human melanoma, where the melanoma's cell nuclei are stayed yellow, the vesicles are red, and you can see on your right that there are large red clusters in each tumor cell. Presumably these are multivesicular bodies. On the other side, the red vesicles are sort of spread in the cytoplasm. So we actually can visualize the biogenesis of these vesicles in tumor cells. Coming back to biogenesis of extracellular vesicles, and at this point I should stop and tell you that this is a sort of a new field, the field of exosomes and extracellular vesicles. And so the nomenclature of extracellular vesicles, it's slightly confused. Exosomes are actually a subset of extracellular vesicles, and these extracellular vesicles are of many different types. So you can see here that exosomes are small extracellular vesicles, and within exosomes themselves, you can see they are different sizes of these vesicles. And then any cell also produces much larger vesicles, which are not formed in the endocytic compartment, but rather bud from the surface of the cell membrane. And so these are microvesicles, which are much larger than exosomes, and they often carry oncosomes, oncogenic proteins. And in addition, there are even larger bodies that cells release, and those would be apoptotic bodies, and these are really very large aggregates of dying, components of dying cells. So all cells produce a heterogeneous mix of extracellular vesicles, and exosomes or small extracellular vesicles are a subset of these various vesicles that cells produce. And so these extracellular vesicles are released into extracellular space, and they circulate freely. And I'll show you that in just 1 minute. They are released by cells which are activated. They are, as I again emphasize here, of various sizes and types, and they are rich in cargo, variety of cargos, and they seem to play a role in variety of important processes which are listed here, and this is from angiogenesis through cell activation, through inflammation, and they also serve as biomarkers, the biomarkers of pathologic conditions or diseases. Now, I said they circulate freely. How do we know this? Well, some years ago, we performed a study to ask whether whether extracellular vesicles, exosomes, have the ability to cross the blood-brain barrier and other tissue barrier. And so this is an experiment where we generated in cell culture and purified exosome from a variety of different cells listed here, including primary T cells, but also murine vesicles, as well as human vesicles produced by human cells. They were all isolated by size exclusion chromatography, and they were labeled with iodine-125 and injected into mice. And then my collaborator, Dr. Banks, determined whether these labeled exosomes were found in the lung, spleen, kidney, or liver, and whether they were all found in the brain. And here you can see result of this elaborate experiment where the injected vesicles travel around, and you can see that they are readily seen, visualized in spleen, liver, lung, and kidney, but also in the brain. And it's very interesting that primary T cells, for example, accumulate very, very richly in liver, in less so in other organs. But at the bottom here under C, you can clearly see that these vesicles